All right, welcome to our next flipped notes. These are going to be on cellular transport. So yesterday we did talk about how the cell membrane, what it was made out of, its structure, and that it was a fluid mosaic model. So I did want to show you this video. I think it demonstrates what fluid mosaic means very well. Maybe not. All right, so I love that video just because I think it really demonstrates everything, and it's very soothing. All right, moving on to our next one. Oops. Technology is not working for me today. There we go. All right, so cells are enclosed within a semi-permeable membrane that regulates their interactions with their surroundings. So how does it do that? That's what we're going to be looking at today. How does it maintain homeostasis? So first, you need to understand the word concentration. I know many of you have heard that word before, but it's the mass of solute, what was dissolved, in a given volume of solution. So for example, if I took 12 grams of salt or 12 grams of Kool-Aid and I dissolved it in 3 liters of water, the concentration would be 12 grams over 3 liters. All right, so membrane permeability. There are 3 types. Impermeable, which means nothing can go through it. It's a block wall. Freely permeable, which means anything can go in and out of it. And selectively permeable. You might also hear that as semi-permeable. So selectively and semi-permeable are the same thing. Our cell membranes are semi-permeable. Some things can go in, other things cannot. The way I think of it is a bar. So there's a bouncer at the door. If you're 21, you can go in. If you're not, you can't. That's being selectively permeable. So we're going to look at some different transport options. The first one we're going to look at is passive transport. That means no energy is needed. And there is, for the first one, which is diffusion. Okay, so diffusion is the movement of materials or particles or solutes from higher concentration to lower concentration. So a little picture right here, you can see as it dropped the purple sugar cube in. It takes a few minutes, but it slowly, as it dissolves, those particles are actually diffusing into the water. Okay, a type of diffusion, it's a special type of diffusion, we're going to talk more about it later, is osmosis. Okay, so osmosis is a type of diffusion where water moves from an area of higher concentration to lower concentration. So again, diffusion is particle movement, osmosis is water movement. The second type of passive movement is filtration. So that's pressure causes the solutes to move across the membrane. We're going to see that in the kidneys. We'll be talking about that more with the urinary system. All right, active transport. So again, passive, no energy. Active requires the cell to use energy. It's usually going to be ATP. There's two categories. We have endocytosis. Excuse me. Endo means in, cytocell, so it's the packaging of extracellular materials. It's bringing stuff into the cell. And then we have exocytosis. Those are vesicles created inside the cell and released outside. Exo, exit, exiting the cell. We see this with the transmembrane potential. We'll talk about this more when we look at the nervous system, but intercellular fluid has a slight negative charge and extracellular fluid has a slight positive charge. That creates a potential difference. Okay, so you can see exocytosis, it's pushing something out. This is going to take energy. Endocytosis, bringing something in, that will also take energy. And the last one we're going to look at for this is carrier-mediated types of movement, and that uses proteins, those proteins that are found on the cell membrane. So there are some limitations to these, and those limitations are specificity. Now I can't speak. I'm tired, you guys. I do apologize. But it only binds certain substances. Only certain substances. So it's very specific about what it's going to bind. Also saturation. There's limited numbers. So saturation means it can only hold so much. Again, I think of it as a paper towel. So in a paper towel, it soaks up a lot of water if you just leave it there, but it can only hold so much. Once it is filled, it is saturated. It can't hold any more. Okay? There are two types of carrier-mediated diffusion. The first one is facilitated diffusion. And so that's a large molecules, they bind and it results in movement that way. So no ATP is used, but once it's saturated, it's done. It can't hold anymore. So facilitated, it helps. It just helps move larger molecules through the cell membrane. The second one, active transport, which we already mentioned a little bit. ATP is used, and an example for that would be the sodium-potassium pump. So intracellular sodium, you have 3 molecules, are exchanged for extracellular potassium, 2 molecules. Okay. All right, so we're going to look a little bit closer at our diffusion and osmosis. So again, a little bit more in-depth definition of diffusion is random movement of particles down a concentration gradient. It goes from higher to lower. So things go from being squished to not squished. So again, think about if I moved everyone to one corner of the classroom and just shoved you guys together. If I didn't keep you there and constantly yell at you to stay in that corner, you would slowly start to spread out, because no one wants to be shoved in a corner together. It's the same with the particles. It wants to move out. It wants to reach something called equilibrium. So this is a short video that demonstrates that. Okay, so yes, it cut off. It was supposed to. We're just looking at diffusion. So again, that was looking at a particle. This one is looking at like food coloring. You have your water, you put a drop of food coloring or dye in there, and again, it's slowly going to disperse until it's evenly dispersed throughout the bottle or beaker of water. The second one is osmosis, and that is the movement of water across a membrane down a gradient. It's called osmotic pressure, and it goes from lower to higher in osmotic pressure. So here's a quick video demonstrating that. Maybe. Sucrose is table sugar. So osmosis, again, is a division. It's a type of diffusion. Diffusion, movement of particles. Osmosis, movement of water. So rules, diffusion of water. It occurs across a selectively or semi-permeable membrane. Water goes to areas with a higher solute or more particles in it. There's three types. We're going to go more into these types of solutions. Isotonic, no osmotic flow. Hypotonic, water flows into the cell. And hypertonic, water flows out. So we're going to go more in depth into these. This I like to show real quick and explain. So if we have some tubing, this is before osmosis. Right here we have a selectively permeable membrane, and as you can see here, let's call these pink dots sugar molecules. There's a lot more sugar molecules on this side than this side. Well, the sugar molecules cannot fit through the membrane, but it doesn't like being uneven. So what's going to happen is the water can fit through the membrane. The water is actually going to push its way through here until it ends up like this. Okay. And that does happen. All right, let's go talk about the different types of solutions. So, I didn't realize that got cut off. Isotonic concentrations inside and outside are equal. That's what isotonic means. It means it's reached equilibrium. So if we're looking here, this is an isotonic solution. Water molecules move into and out of the cell at the same rate. It's never static or not moving. It's always moving. It's just equal. It's equal on the inside, equal on the outside. So things go in and out equally. The second type of solution is hypotonic, hypo below. So hypotonic solutions, the concentration of water inside the cell is lower than outside the cell. So what happens if we're looking in a hypotonic solution, water enters a cell by osmosis, causing the cell to swell. So if we look in here, there's a lot more sugar on the inside, less water. On the outside, less sugar, more water. So the cell, again, doesn't like that. So we have a lot more water coming in. Some is still going out because, again, things are constantly moving. So some in, a lot in, a little bit out. And then our third one is going to be the opposite. That's hypertonic. Remember, hyper means above. So the concentration of water is higher inside the cell than outside. So in this case, you can see we have the little water particles, we have our sugar particles. There's more water inside, less water outside. And the cell doesn't like this. So what happens is the water leaves the cell by osmosis, causing the cell to shrink. So a lot of water leaves, very little comes in. We see this in the body with our blood cells. So again, our cells, our red blood cells are in blood. Blood is an isotonic solution. The concentration of the solutes in the blood is the same inside the cell as and outside the cell. If we took red blood cells and we put them in distilled water, there's not many particles in distilled water. There shouldn't be any particles at all. So what's going to happen is water is going to rush into them, and it will actually cause the cells to swell up and blow up. Okay? That's why you cannot inject water into your veins or arteries. So if you're dehydrated, what they actually put in an IV is a glucose solution so that it's isotonic and it replenishes your body that way. Okay, so this would be hypotonic. Solution has a lower solute concentration than the cell. On this side right here, it's hypertonic, so cells placed in concentrated salt solution causes the cells to shrink. So the solution has a higher solute concentration. There's more salt outside the cell, and that causes water to rush out of the cell. That causes your dehydration and why you get thirsty when you eat salt. So again, key notes, things you just need to remember, that your body likes to even things out. It wants to be in homeostasis. So unless something like the cell membrane prevents this from happening across a freely permeable or water permeable membrane, diffusion and osmosis will quickly eliminate concentration gradients. So again, that just means if something is out of whack, it's not even, diffusion and osmosis will quickly try and make it even. And thank you for watching our cellular transfer.